day, everyone. For this presentation, I will be reporting about the green algae Haematococcus pluvialis. For the outline for today's presentation, I will start off by discussing the hierarchical classification and phylogeny of the said species, followed by its illustration or morphology, and lastly are the things that are quite interesting about Haematococcus pluvialis. Of course, before we proceed to the description of the algae, it is first important to know its hierarchical classification. Haematococcus pluvialis belongs to the phylum Chlorophyta, the class Chlorophyceae, the order Chlamydomonadales, the family Haematococcaceae, the genus Haematococcus, and species Pluvialis. In a paper by Lin et al. In 2017, a genome skimming method was used to deduce the complete specific rRNA repeated sequences as a nuclear marker, which is used as a molecular tool to reveal the relationships between green algae. Highlighted in black, the results showed that Grella ellipsoidea is closely related to Myactinium raceri, while Haematococcus pluvialis is closely related to Chlamydomonas sp. In contrast, the freshwater macroalgae Agragopila linnae was shown to be phylogenetically distinct from Chlorella ellipsoidea, Haematococcus pluvialis, and other green algal species tested in the study. For the description and morphology, Haematococcus pluvialis is a freshwater unicellular green algae that is microscopic and can produce their own food through photosynthesis. It is ubiquitous as it is widely distributed in many habitats worldwide. It is also able to form cysts, which allows the species to be well suited for survival under extreme conditions such as radiation, temperature, and salt concentrations. Compared to other algal species, H. pluvialis has a rather complex life cycle, which consists of four distinguishable cellular morphologies: the macrozooids, microzooids, palmella, and hematocysts or aplanospores. Macrozooids predominate under favorable culture conditions in the early vegetative growth stage. They are spherical, ellipsoidal, or pear-shaped biflagellate cells presenting a characteristic gelatinous extracellular matrix. Under unfavorable conditions, cells start to lose their flagella and expand their size, developing into non-motile palmella. However, when stress conditions prevail, palmella transform to asexual aplanospores and become resistant to extremely harsh environments. As seen in the figures, mature aplanospores are red in color because they accumulate large amounts of secondary carotenoids, which will be discussed in the next slide. H. pluvialis is an interesting species as it is a promising microorganism that now showed potential to be a nutraceutical for human health because of the ability to produce astaxanthin, which is used as a coloring agent for aquaculture, as a protection for the skin against UV-induced damage, for tumor therapies, and for prevention of neural damage associated with age-related degeneration. H. pluvialis has developed into an organism that can be cultivated on an industrial scale. In addition, H. pluvialis can also generate chlorophylls A and B and primary carotenoids compounds, namely neoxanthin, violaxanthin, zeaxanthin, lutein, and beta-carotene, which suggested their great development and commercialization. And that ends my report about Haematococcus pluvialis. Thank you for listening.